Okay, so I just wanted to take a couple minutes to go through this genetic traits lab, just because I thought there might be a couple areas of confusion. I have kind of rewritten this section here, not rewritten, reorganized this and put these little letters here because these are all going to be traits that you will fill in on this table below. And so then I just took a screenshot of this and kind of pulled it out to the side so I could keep it up while going through this table. So I would just kind of like keep this by your side because this is really all you need to be able to fill in the table. They tell you exactly what genotypes are associated with each like color of cat that you're looking at. So I just chose a few cats to go through now with you just to kind of give you a couple examples of how to do this. So if we look at Luna here from the Humane Society, she is obviously a female. Females always have XX for their 2 chromosomes, for their sex chromosomes, and then looking at the genotypes for the rest of these. And remember that genotypes is, like what alleles make up this certain trait that we're looking at in an organism. And then the phenotype is what that trait looks like physically on that organism. So looking at white alleles first, if they are all white, they're going to have a big W and then either another big W and a little w. If they are any other color, even partially any other color, they're going to be little w, little w. So Luna's going to be little w, even though she is pretty, a good proportion of her is all white. She does have other colors. So what does that tell us about the white allele, white trait? Is white going to be dominant or recessive? So hopefully you said dominant because an all white cat would have a big W and then either another big W or a little w. We can't know, even if we have an all white cat, what their entire genotype is without actually doing some genetic testing on them. So tabby coloring, and this is why, like when you have a litter of kittens from 2 parents, even if the parents are both orange, you can have just such a wide array of kittens because there are so many genes that go into the coloring for organisms. And if they are heterozygotes, mom and dad, then they can produce offspring that look different from them. Okay, so for the tabby coloring, if the cat is tabby, they're going to be a big A because tabby is dominant, that coloring. If they're a solid color, there'll be little a, little a. So Luna has some tabby coloring in her. And if you actually click on her picture and scroll down to the bottom, they will say white and gray tabby. And you don't even really need to look at the description. You should just be able to kind of recognize what tabby is. So on her tail and on the sides of her face, you can see that striped coloring. So she's going to be a little a for the tabby genotype. Oh, sorry, big A, little a for the tabby genotype. For orange, let's see. So orange and black is what we're looking at here. And they're both found on the X chromosome. And again, this is why female cats can be calico colored like Luna here is. That means that she has both the orange and the black alleles. And so that's actually another example of co-dominance, like blood typing. If you can express both things at once and none of them are covered up by the other, so it's not like if she has the black allele that it covers up the orange one and it just makes her black. She can be both orange and black at the same time. So that's a good example of co-dominance, and that might be, potentially a test question or a lab practical question, the idea of co-dominance for female calico cats. So she would have the allele for orange on one of her X chromosomes, and her other X chromosome is going to have the allele for black coloring. So calico or tortoiseshell females. Luna is calico. Tortoiseshell is when they are primarily black and have some orange in them. And then this last one, the dilute locus, which can be a little bit confusing. I have Tigger up here next to Luna because I think that she's really, really easy to see this dilute coloring in. So Tigger would actually be dilute for both orange and black. And you can see how kind of light gray and light orange her coloring is. Luna is very similar. You just can't see it on her whole body. So she is going to be dilute. And it doesn't really matter if we're talking about gray or orange. If they have the dilute genotype, then they are going to be dilute kind of for whatever color they express. Even though you do see some darker black down in the tip of her tail, we're going to call her dilute because mostly she seems to be a light gray and a light orange. Okay, Claire, who's a cute little tortoise shell. Again, obviously a female, so 2 X chromosomes. What about her white hair allele? Obviously, she doesn't really seem to have much white at all in her. And it doesn't really matter if she has white in her. If she has anything other than white, even a bit of it, she is going to have 2 of the recessive alleles for the white genotype because white is going to be the dominant genotype. Okay, so the tabby coloring, I would say no. She looks like she is calico, but we don't see any striped appearance on her body. So if we don't see any stripe, like a tiger striping in their fur, then we can just assume that they are not tabby. So we would call her, even though she's tortoise and that calico mixture, each of her individual hairs are a solid color. There's no striping on individual hairs. So we will say little a for solid color. And in the orange and black, again, she's going to be just like Claire, where she has 1 of her X chromosomes with an orange allele on it, and then 1 of her X chromosomes has the black allele on it. And then dilute, does she look diluted in her orange or black fur to you? I would say no. So I would say that she has 1 of the big D alleles. The other one we can't know. She could have a little d allele on the second one, but we can't know. If there is any dilution occurring, that means that they are recessive, recessive homozygote, as in little d, little d. If they have any solid black or solid orange without diluting, they have to have the big D allele. So think for a second, what does that mean about the dilution genotype? Is dark coloring or bright coloring going to be the dominant trait, or is the dilute coloring going to be the dominant trait? So they can only be dilute if they have little d, which means that dilution is recessive. So if they have any big D alleles, they will have that really bright orange or black color. So this is complete dominance. It's not like if they have big D, little d, they'll be somewhere in between for dilution. It's just either diluted or not diluted. So complete dominance there. Let's go and look at Gary. I think he's on page 2. Makes me want to go and adopt another cat. I think Nova looks like he'd be a really cute cat to have. Okay, Gary, I chose him because he has a funny picture. Let's look at Gary's description. So he is an orange tabby. And so I think that that's obviously very clear from his picture. And you can kind of take a second to think, is Gary dilute or not dilute? What would his other genes be? And if you want to pause the video here, try to fill it in all yourself, and then I will list out the answers. So Gary is obviously a male. Does he have the white hair allele? Remember, if they have any color on them, white is dominant. So if they have any color, they have to be homozygous recessive. Tabby coloring, definitely he is a tabby. We don't know what that second allele is because tabby coloring is dominant. So we can't be for sure on what the rest of his is. So males have X and Y chromosomes. So you have to carry that over to sex-linked traits. So there is not going to be any allele on the Y chromosome. So if you have an orange male, you are going to have X, the X chromosome with the orange allele on it, and then a Y chromosome. And this is why you can't have a male who is a calico cat, because they only have one X chromosome. You could have a male cat who had Klinefelter syndrome, and then technically you would have a male cat who has calico coloring. if they had XO, XB, and a Y chromosome, but that would not be a normal genotype for a male. That would be a situation where you had non-disjunction occurring and you had two X chromosomes, which is not a normal situation. Diluted or not diluted, if orange has been diluted to cream, and it's hard for tabbies, but I would say that he does look fairly light coloring, just because I have had orange cats that look much, much darker or brighter than his orange is. So in my opinion, I would go ahead and say that he's dilute. I'm not really going to check this for correctness, like do you know your cat fur really, really well, but do your best to fill in this table with cats, and you can use a couple of mine if you want to, but then just make sure you fill in this table completely. Once you have filled in this part, then you can come down here and say which was the most common phenotype for white coloring. You know, by the looks of it, there are not that many all white cats in the world. I have had an all white cat, but they're not super common. And so the most common phenotype for that is going to be some color, or not all white. The most common genotype is going to be little w, because the white allele would be dominant. And so if you had an all white cat, it would be big W. The least common genotype, I would probably just say being white would be the least common genotype, and being an all white cat would be the least common phenotype. For tabby coloring, I would say tabby is probably going to be kind of a toss-up. So you can kind of fill in your table and just select which cats you think are cutest or whatever. I don't care. If it ends up being a 50-50 split, you can say that in your table. If you end up using more tabby cats than solid color cats, then go ahead and fill it in with whatever results you get. Same thing with orange and black and dilute coloring. This will probably depend on if you choose mostly females or males, because obviously the females, if you choose females who are calico, that could kind of affect your results here. But just based this on your results, there really isn't any right or wrong answer. So for the human genetics part, this website, NCBI, and if you go into anything human related, this is really the primary research site for human research. So you can always come here instead of like a library or website or Google Scholar to find a lot of scientific articles. So you're just going to use this website, look up each trait in the data and observation sheet, find a result that kind of talks about the trait that you're looking at. So which chromosome is it on? They will probably just list out the chromosome that it's on, and then what is the gene called. Hopefully you can find all of these things pretty easily, but I'll go through an example on here. So here is your table that you're going to use to fill this out. So if I just look up Turner syndrome, let's see. So I have looked around for a little bit. I don't think that there is actually a better one than this first link. So it won't always say it in the title of the link that you're clicking on, but it should say in bold terms below if you're on the right syndrome. Most of these are fairly easy to find, but I'm going to help you go through the Turner syndrome one, because this one can be a little bit confusing just because there's a couple different possibilities for Turner syndrome. It can be completely missing the X chromosome, one of the X chromosomes, or it can be a partial deletion of the X chromosome. So the wording of it gets a little bit confusing. The location, this will either be an X or a Y, or it'll be a number here, which will tell you the number of chromosome that you're looking at. So the sex for Turner syndrome, and a lot of these, if you're having trouble finding that piece, just Google it or go to our lecture PowerPoint, because the lecture PowerPoint has a lot of those details. So the lecture PowerPoint from, I think it was module 5, the first one. So this Turner syndrome, I believe, would be female, number of chromosomes. So you could say 45. Most humans have 46 chromosomes. So if we're missing a whole X chromosome, obviously you would have 45. How are the chromosomes abnormal? Missing an X chromosome or partial X chromosome would be the description for Turner syndrome. What are the symptoms? So you can kind of find quite a bit in this paragraph, but it's a little bit confusing just because they have a lot of jargon. So Turner syndrome is associated with a characteristic of neurocognitive profile that includes impaired visuospatial, so perception, visual and perception impairment, and then usually cognitive impairment as well. And I will tell you that a lot of times they will say mental retardation. And I know that for some people that word can kind of be a trigger. It is totally scientific in this context. So it's just, you know, just don't think about the negative connotation with it in society. You will see a lot in science though, because it is just kind of a scientific term, not meant to be a description of a person in any sort of way. So there is also a PowerPoint associated with this assignment. So if you come to the Lab 10 assignment online and you download the Word document, just keep up this screen so you can go through and really kind of quickly get the genotype mishaps, or I mean the chromosome mishaps on each of these. So normal female, obviously 2 X chromosomes at number 23. You can see all the other ones, how they look there. Normal male here with an X and a Y. Turner syndrome on this case would be missing a complete X chromosome. It is not always completely missing. Cru-du-Chat syndrome is, or Cru-du-Chat, because it is French. If you kind of hunt in these chromosomes, you will find that on number 5, chromosome number 5, we are missing a partial piece of that chromosome. So if we go back to the website here and we just wanted to look for Cru-du-Chat syndrome, usually it's just going to be the first result, first hit. So we're not getting that table loading correctly, but it will usually at least kind of tell you where these things are. So short arm of chromosome 5, and then they usually give you clinical features down here. So Cru-du-Chat means cat cry in French, and so these individuals are going to make like a high-pitched cry on kind of like a normal or like a regular basis, which is why it's called that, but they will have some other symptoms as well. And so you can just choose like a couple of the big symptoms. And if you want to, again, go back to our lecture PowerPoint to help you answer that, that's totally fine. So you can go through this PowerPoint first and kind of write down what you think the issue is with the chromosomes, double check it with the website, and then try to find you know, a couple details about each one of them, and that should really be good for that. So there are quite a few diseases, and then there is one question at the bottom here, so just don't forget to answer that question. I will try to have these things graded for you before you take the lab exam on Wednesday. And remember, that is going to be in class on Wednesday. I am finishing up grading the 2 labs that you just did last Wednesday. And so I will send pictures of those to you. But the sooner you get in all of your assignments for the past couple of weeks, the sooner I'll be able to grade those and get them back to you before you take the exam on Monday. So just kind of stay on top of getting these things turned in on time so you can have helpful feedback before the exam and understand kind of what you missed so you don't do the same things incorrectly on the exam as well. So with that, have a great rest of your weekend, and let me know if you have any other questions over this material.